everybody. Coming at you from my office. We're going to go over the central nervous system to start our new unit in physiology class. For this lecture, we'll go over the brain. And one thing I'd like you to do is to go to Canvas and look at the module there and look at the background on the central nervous system and the brain. Much of that is what you learned in anatomy if you took anatomy, and if you didn't, then you can learn it there, and it's also a good review for those of you who took anatomy. So from now on, make sure you bring your brain. There it is. And let's get going. Let's talk about the central nervous system and the brain. Right, so one thing to remember is that the central nervous system consists of both the brain and the spinal cord, because these are the areas where integration is going on. What it means is sensory information is coming toward these areas, and these are the areas then that can make decisions about what type of motor response to create or what effector organ to stimulate. Okay, and so both the brain and the spinal cord have the capability to do that. So they're both very important for the maintenance of homeostasis. You saw that already in one step of the homeostasis was the integration center, okay? And so that's where this is happening. So you'll read in a lot of sources that the central nervous system, or at least the brain, contains 10 to the 11th neurons, which means 100 billion. You can see I crossed that out because some recent research was done and showed that the human brain has actually 80 billion neurons in it. So now we know. The difference of 20 billion neurons is quite large. So it's actually, we need to keep with that accuracy. 20 billion neurons is about the size of a monkey's brain. So like a baboon. So that's a lot of tissue. Alrighty, so now we know. But look at all the synapses. So you learned in the previous unit that a neuron can branch and synapse with different cells. And so there are about 100 trillion synapses as well in the central nervous system. So this is responsible for everything, how we perceive the world, what we do, what we feel, what we think. So in the Canvas shell, I listed there many of the protections of the brain, things like the meninges, the physical protection from the skull. I mentioned the cerebral spinal fluid. This is another protection of the brain. I wanted to spend just a little bit of time on it because it's very important. So the blood-brain barrier. The blood-brain barrier prevents materials coming from our general circulatory system from leaking into the brain and affecting the neuron cells there. And that's really important because we don't want our neurons to malfunction. So if you recall from the cell signaling lectures, that cells can signal one another or communicate with one another through different types of molecules, such like paracrines, hormones, that kind of thing. And those hormones travel through the blood indiscriminately, so it would travel up toward the brain. And so hormones then could affect the neurons in different ways, and the brain may not want those neurons affected. Okay, other toxins, things like that can come in. So let me zoom in on this picture for you. So what we see is that the blood-brain barrier occurs because of specific cells, such as astrocytes. And what they do is their projections, that's a glial cell, those projections kind of flatten out and lay over the surface of the capillaries that come into our brain to provide the blood and the oxygen and other things that we need. Most capillaries are very leaky because we want nutrients and oxygen and hormones and other things to leak out and get into the interstitial fluid, but not in the brain. Again, we want to be really careful about controlling what gets in there. So there are tight junctions in between the cells that make up the capillary. Those cells are called endothelial cells. It's a type of epithelial cell. It is an epithelial cell, But when we refer to epithelial cells associated with the cardiovascular system, which capillaries are, um, we refer to them as endothelial cells. So you'll hear that again when we do the cardiovascular system. Okay, and so you can see a cross section there. You see with the tight junctions that forms a seal so things can't get out, makes a leak-proof barrier. And then the astrocytes are there because anything that does get through via osmosis or diffusion through the membrane, it will end up going through the astrocyte first. And then the astrocyte can control what actually ends up in the brain tissue itself. So that's the blood-brain barrier, very important in our for our brain's protection. The other thing that you will see when we look at the brain is that the brain is highly organized. And one way that it's organized is through something called gray matter and white matter. And really all this refers to is the part of the neurons that are clustered together. So the neurons tend to be clustered, the cell bodies tend to be clustered along the outside of the brain, and that's what we refer to as the gray matter. Let me skip to this slide real quickly, and you can see that. You can see in this picture of the cross-section of the brain in the upper right, that that outline of the gray along the outside, those are the neuron cell bodies that are clustered in those areas. So we call that the gray matter. That's where processing occurs. So the neurons bringing sensory information will synapse with the cell bodies in that area of the gray matter. And then it's those cell bodies that send a motor response outward. Okay, the white matter that we referred to here, so you see on the gray matter, that's the neurons, the cell bodies, and the dendrites are also there. But the white matter then are the axons from those neurons. And the axons, remember, in the CNS are wrapped in cell membrane from cells called oligodendrocytes. In the earlier unit, we looked at axons that were wrapped in the peripheral nervous system, and that happened by a cell called a Schwann cell. So this is different in the CNS. These are the oligodendrocytes that wrap them, but it performs pretty much that same function of allowing electrical activity to pass more quickly. So that electrical activity, those axons pass through the brain itself, and those axons bundle up together and they form that white matter on the inside because the oligodendrocytes tend to have more of that white coloration to them. Okay, the lipids is what it is, making that white color. If you look down to the lower left side, the cross section of the brain, those lines are showing the white matter and how organized it is. So the axons making the white matter are arranged and traveling toward different areas of the brain in order to make all of these different connections. So you can see there can be short connections from very short areas of the gray matter to another area of the gray matter. It can go completely from the front of the brain to the back of the brain and vice versa. And then if you look in the lower right side, you also see from a frontal view of the brain, those lines there are indicating the axons, the portion of the white matter. It's coming from the spinal cord up to the cerebrum and vice versa. And it's also, they're going from one side of the cerebrum to the other side. So a lot of connections that are going on there. Okay, those are referred to as projection fibers. Okay, sometimes they're referred to as association fibers. So we might hear that later. And also in the upper left corner, you'll see the arrangement of the gray and the white matter in the spinal cord. It's opposite of what it is in the brain, because in the spinal cord, the gray matter is in the center of the spinal cord and the white matter is on the outer portion. It's the cell bodies in the spinal cord are in the center, and the axons then move through the length of the spinal cord on the edges. So that's the opposite of what you see in the brain there. Okay, so gray and white matter occur in both the brain and the spinal cord. Those are both the CNS. So we'll look at that in the spinal cord in the next lecture. Okay, so with your brain yourself, again, make sure that you review the different parts of the brain and what their functions are, what their overall functions are. I'm going to focus on, since you did a lot of that in anatomy, I'm going to focus on some more specific parts. So overall, you will look at the cerebrum, and that's the most of the part of the brain that you can see there. It's separated into the right and left hemispheres, and we're looking at the picture on our left is the forebrain, so we're looking straight at the brain. That means it's another word for the cerebrum as well. The outer portion of the cerebrum is the cerebral cortex, and that's what you saw over here with the gray matter. It's the gray matter of the cerebrum that forms the cortex. And then from there, we'll look at the inside of the brain in a little bit, but on the inside, we'll see portions inside of the white matter of the cerebrum that are where gray matter is embedded. Those are the basal nuclei. We'll also look at the diencephalon, which includes the thalamus and the hypothalamus. We'll look at the cerebellum and the brainstem, along with included in that is the midbrain, the pons, and the medulla. So again, make sure you know all of those parts, and we're going to go over some of those parts today. So first we're going to start with the cerebral cortex. Again, this is the outermost portion of the brain. It's a very thin, convoluted layer of gray matter. Convoluted means it's folded. So if you remember from anatomy, those foldings, when they project upward, that's called a gyrus, or there's many gyri. And then when they kind of fold inward, that inward folding is called a sulcus, or many sulci, right? So it's in this cortex, that gray matter, where we have the highest level of neuroprocessing. This is where right now I'm deciding what words to say. Okay, it's helping us perceive our environment. It's helping us to formulate ideas. This is where how we recall past events. This is how you control your body movements, your voluntary body movements. Okay, so this is a very complex integrating center, and it's actually very well organized. Again, not just between the gray and the white matter, but even within each of those. So you saw the lines of the white matter running through. Here it shows the gray matter. So there's actually 6 well-defined layers of gray matter, and it's based on varying distributions of several distinct cell types. You don't have to know each of those cell types, but under B there, that picture is kind of showing the 6 different layers and the different types of cells and how they project through the layers. So the layers are defined by the accumulation of the cell bodies in those layers. Okay, the layers get organized into functional vertical columns, and it extends perpendicularly about 2 millimeters from the cortical surface down through the thickness of the cortex until it reaches the underlying white matter. Okay, so the neurons within each column can actually form a team. So each cell is involved in different aspects of the same activity. So for example, this perceptual processing of the same stimulus from the same location. Okay, functional differences then come from different layering patterns within the columns. So for example, in areas for sensory perception, layer 4 that you see there is expanded with more of a type of cell called stellate cells. These are important in processing sensory information. But in areas where we control our skeletal movement, there is a thickened of layer 5, which contains the large neurons, which are called pyramidal cells. Again, you don't have to worry about the names of those cells, but I just wanted to give you an idea of that organization, how highly organized our brain is, even within that 2 millimeter thick area. All right, and this is what it would look like under the microscope. So you can kind of see the dark spots are the cell bodies and all of that organization within columns and things. Okay. The cerebrum is also separated by different lobes, and these are functional areas of our brain, of the cerebrum. So again, you learn this in anatomy. Please review it. Review what each of these lobes are and their general functions. And some of these lobes, I'm going to get to more specific functions. We're also going to piece together how these lobes interact with one another. Also pay attention to the central sulcus. That's a main sulcus that separates the frontal lobe from the parietal lobe. And that's going to be very important because the gyrus just the front, in front of the central sulcus, and the gyrus just behind the central sulcus, they're going to be very important functional areas. Okay. And then another portion of the cerebral cortex that you may or may not have gone over in anatomy. Some instructors do, some don't. But you can see this is a portion of the cerebral cortex where the parietal lobe folds inward before the temporal lobe begins to form, right? So that folding inward right at the base of the parietal lobe, that's called the insula. And that is a functional area in and of itself. It's not really part of the parietal lobe or the temporal lobe. So it plays a role in memory encoding, and there's a lot of research going on right now about exactly what the insula does. Okay, so it says there it integrates sensory info with visceral responses. So visceral responses are your organs in your abdomen. Okay, and it can also coordinate cardiovascular responses to stress. And we believe it plays a role in the awareness of things like smoking urges, cravings, and anxieties. So that's one reason that this area of the brain is beginning to get a lot of attention, so we can understand those processes. Okay. So here we go. I mentioned the central sulcus. And so here, the top picture there, you're looking down from an aerial view, looking down on the top of the cerebrum. So you can see that the cerebrum is separated into the left and the right hemispheres, right? And so you have that fissure right down the middle. But then going in the perpendicular direction, you see the central sulcus, and that separates the frontal lobe toward the front of the brain, which from our view is the top, from the parietal lobe that is toward the back of the brain, not the way back, but just behind that central sulcus. Okay, and so the two gyri just in front and behind of the central sulcus are very important areas. These are where neurons are organized in order to, in the blue area, part of the parietal lobe, where we receive sensory information. And then in the kind of the, I don't know what color that is, peachy color, that is indicating the the motor cortex. And so that is a collection of nerves that controls our voluntary skeletal muscle movements. Okay, this is where we control that. Now the pictures below them are referring to something called a homunculus. Homunculus literally means little man. And what it is, you can see, is that it is showing the distribution of motor output. So if you look over down to the right, that's the distribution of motor output from the primary motor cortex to the different parts of the body. Okay, but notice that the little person there, the little man that's drawn on there, is distorted. So the distortion of the graphic representation are the body parts that indicate the relative proportion of the primary motor cortex devoted to controlling skeletal muscles in each area. So for example, as humans, we are we use our hands a lot. We really rely on our hands in order to manipulate objects and things. So right now I'm talking about motor. So we use our hands to grab onto things and um, to do a lot of things, use tools, things like that. So you can see on the little person that the hand area, that portion of the brain devoted to controlling our hands and our thumb, look at how big the thumb is, right? We have opposable thumbs. And so that distortion of the hand being very large indicates that we have a lot of neurons devoted to that. Okay, I can show you just showing you the motor area. So again, the hand looks really large. Our legs and knees and ankles are important, but not as much as our hand. Okay, so this is relative to maybe a different organism, right? Look at the distortion of our mouth, very large. We communicate by talking and using our mouths, right? The tongue is very large. It's very important in communication. So again, the relative size of the homunculus, it's kind of a map that indicates how many neurons are devoted to that motor movement. The same thing is true of the sensory portion. So on the primary, what's called the primary somatosensory cortex, that's the gyrus just behind the central sulcus. So that's mainly found in the parietal lobe. But again, this is now talking about, refers to the sensory that we put into, that is important to us as humans. So our hand is very enlarged again. Again, we use our hands for sensory, to touch, to determine what things are. Okay, so that is enlarged. Our lips, we use our lips to test different foods and things like that. And so that's more of a sensory thing there, right? You can go on and see kind of the different things that are important to us. Okay, so again, the sensations come from the body surface, and that's so those are things like touch, pressure, heat, cold, pain. Those are things that refer to as the somatic somesthetic sensations. Okay, it's also what's known as proprioceptive input. So that's the input coming from our joints. It provides information to us to know where our body is in relation to the external environment. Are you standing? Are you leaning? Are you standing straight up? Right? Is your elbow bent? Is it straight? Those are proprioceptive input. Okay, so it gives you an idea of your relation of your body position. All right. So I wanted to show you a difference so you can see the human homunculus. And so I threw this in here so you can, what if you were a star-nosed mole? So a little guy under A there, that is an actual star-nosed mole. That's his anatomical proportions. There may be about, I don't know, they can get up to maybe 5 inches long or something like that. So this is an animal that burrows in the ground and mainly lives its life underground. Okay, and so living underground, it doesn't really have the ability to see much of anything. So its eyes are very well reduced. But that projection on the nose, when he's burrowing through, he's actually feeling the soil in front of him. And he can tell, am I running into a rock or just smaller pebbles? Are there insects in front of me? So that distortion on the bottom shows how important the sensory information coming from those projections off of his nose is. His front legs, they're digging, they're burrowing through the soil all the time. So those front legs are also distorted in the lower picture to show that how important the motor movement of digging is. And you can see the back legs aren't very different. So he's not using his back legs as much as he's using his forelegs. Okay, I couldn't find a picture of the homunculus over the brain, but this just kind of gives you an idea of how different organisms will be with the different homunculus. Okay, so we're going to talk about the thalamus a little bit later and more in depth, but I do want you to be aware that even when we have simple awareness of the sensory information coming in, touch, pressure, pain, all of that information comes through the thalamus first. Okay, and what it does is it makes you aware of the input that's coming in even before the cortex, the somatosensory cortex, um, can determines exactly what it is, right? So you may have something in your hand, and the thalamus is detecting that, hey, this is something wet and cold. And by the time then, so it just kind of wakes up the neurons towards your um, primary somatosensory cortex, that gyrus there, and when I say it wakes them up, it's bringing them closer to threshold, right? And so now you'll have a quicker processing of what that wet cold thing is, and you determine, oh, it's an ice cube. I'm holding an ice cube. Okay, so it just gives you like a fuller sensory perception, and it helps to localize the source of sensory input, and you perceive that level of intensity. Okay, it helps with spatial discrimination, and then discerns shapes. So that wet and cold thing, you'll even determine it was a cube. Ah, then it's your primary sensory cortex that determines this is an ice cube. Okay, and we'll go over this in more detail as well. But if you're feeling something on the right side of your body, those neurons then that come from that area, they come into the spinal cord, and as they move up the spinal cord, they actually cross to the left side and stimulate the left side of your brain. So sensory information coming from the left side of your body goes to the right side of your brain. Okay, for the most part. The same thing will happen with motor. So the right side of your brain will control the left side of your body and vice versa. Okay, so we'll talk a little bit more about the thalamus and this what's called lateralization as well. So how do we know all of this? Well, some of it comes from the images that we can take of the brain now. So an MRI, magnetic resonance imaging, can actually show brain functions. So what they do is they might prick your finger, your thumb, and the MRI will show the image of where that neuron is lighting up. So it actually shows where the neuron is located in the brain that's controlling your thumb or receiving sensory information. And vice versa for the control of the thumb. You can stimulate a part of the brain on the motor cortex, and it will make your thumb move, right? And with all the other fingers and all the other sensations. That's one way that we learned about that homunculus, to be able to map the brain. One way with this MRI can do that. Some of these are early research came from actual brain surgeries. The brain does not have pain receptors in it. And so when you do a brain surgery, of course, the pain receptors are in the integument and the skull, and so they numb that. But the brain itself, you can stay awake during a surgery. And so therefore, they often do that to make sure that they're not harming any other areas. So they'll have you talk and move around just to make sure they haven't harmed any of these other functional areas. So you can Google on YouTube and see people in brain surgeries, and they're playing guitar and things like that. Another type of imaging, here's a couple of them, a CT scan, and that actually visualizes the soft tissues, so it almost looks more like an X-ray, but it can go through different layers. And then the picture I have here is showing a PET scan, positron emission topography, and this is what measures kind of the blood flow and the metabolism of certain areas of the brain. So when you're being imaged by a PET scan, these areas of the brain will light up in the image because blood flow is going to that area. So you can see here when they play a tone for somebody, they're here, you can see that area of the brain lights up. That's the temporal lobe. That's why we know sensory information for hearing comes to the temporal lobe, right? Sensory information of seeing goes to the occipital lobe, okay? And then you can see speaking has a different area. We're actually going to study that area in a little bit. And then thinking happens much in the frontal lobe, so where you have your thought processes and things. So I wanted to throw those in just so you might, as you go into your careers, you might then see these different types of ways to visualize the brain. Okay. Here's another, the cortical association areas. So these are areas separate from those main gyri that I've spoken about. And so you can see these areas are kind of separated out here. So for example, in the green in the back, the very dark green, that's the visual cortex. But the lighter green just in front of it, or the duller green, that's the cortical association area. So basically, again, it requires input from the primary cortical areas for integration. So if you're seeing something, the optic nerve will bring that information to the primary visual area, the dark green in the back. And then axons through association fibers are connected to the association area, and it's there where some more integrating is going on. So the primary visual cortex decided what you were seeing, but the association area is providing more of a context to what you're seeing. Okay, is it something important? Is it something you need to run away from, right? And so it's going to connect you to other areas of the brain. If it's something to run away from, then that is going to have a neuron fiber that heads to your motor cortex. Okay, so again, association areas. Okay. And here's that brain lateralization. So this is what I kind of mentioned before. This example now, you can see it with the motor cortex. So you see motor cortex, the cell bodies start up in the motor cortex area, in that motor cortex gyrus. And then the axons bring signals downward through the white matter, through the midbrain, through the brain stem, all the way out the medulla oblongata to the spinal cord. And at some point it crosses over from the right side to the left side. And sensory, said again, we saw that go in the opposite direction. The sensory, the input starts in the peripheral area, and then those axons traveling through the spinal cord are bringing the action potentials upward through the spine, through the brainstem, to the appropriate area for that input to come into the brain. Okay, so again, lateralization means the left side controls the right side and vice versa, or the left side perceives what's happening on the right side and vice versa. And that's actually happening because these axons are crossing from, they might start on one side of the body, but they end on the other side of the body. All right, and I also wanted to point out that I mentioned this a little earlier, but again, there are two hemispheres of the cortex. We have a left and right side. And normally the two hemispheres complement each other, but they do have some functional differences. So you can see those differences there. Left cerebral hemisphere is more where you have analytical thought, logic, language, right? This is what's being stimulated right now with science, okay? Right brain functions are more of the creativity, imagination, intuition, holistic thought, right? But again, they complement one another because science can be very creative. You need an imagination in order to look at the world in different ways and to develop experiments and things like that. So they do complement one another, okay? Usually the left side is most commonly dominant, the dominant hemisphere for fine motor control, and that's why most people are right-handed. There used to be a time where we thought handedness showed that if you were left-handed, that you weren't very smart because then you're right brain dominant. We know that's not true today, but that's much to the detriment of a lot of people were actually punished for using their left hand, and they wanted to force them to be right-handed, thinking that was the quote-unquote normal and would help their brain development. We know that's absolutely, unequivocally untrue. As my brother will tell you, my brother is left-handed. I have other comments on that, but that's brother-sister stuff. I also put at the bottom there that TED Talk. It says, Just go to the tedtalk.com and look up Jill Bolt, My Stroke of Insight. This was an actual brain researcher that went through a stroke herself. And so she was able to remember what was going on when she was having the stroke. And it gave us a lot of insight into the functional difference between the left and the right side of her brain. Basically, during her stroke, the left side of her brain stopped functioning, and she was really living through the left side. So it's really interesting TED Talk. If you have some time, check that out. Okay, the next portion I want to go over with the cerebrum is the cortical pathway used for speaking a word that is either seen or heard. And I want to go over this because this is just going to be one example that's going to tie together how information comes to different parts of the brain. Okay, and so this figure's in your book. It's a great one. So there are two areas of the brain that I want to highlight over here, and they're both found only on the left side of the brain. And that's Wernicke's area and Broca's area. And those are both boxed out in the picture here. So Wernicke's area is where you have a plan, you plan the content of a spoken word, right? And so it's more of kind of comprehending and being able to describe objects that are seen, right? Broca's area then is where, when you want to actually formulate those words, Broca's areas is where you determine the sequence of motor neurons that have to fire to get you to move your mouth and your lips or your tongue in a certain way to actually formulate those words. Okay, so let's put this all together. Okay, this is about a pathway for speaking a word that is seen or heard. Okay, so again, the information comes through your eyes, and if it comes through your eyes, then that information stimulates your primary visual cortex, right? If you hear it, that information from your ears comes through your temporal lobe, and that's the primary auditory cortex, okay? Again, visual cortex is in your occipital lobe. Right? So that information coming from either the visual cortex or the temporal cortex goes to the association areas, and those association areas pass it to the one in blue there, number 2. It's called the angular gyrus of the parietal temporal occipital association complex. That's where all this stuff kind of merges. Okay, and it integrates the inputs of sight, sound, touch. Okay, so it's integrating all that. Right here, we're really talking just about sight or sight or sound. Okay, so then if you're going to speak about something heard, the brain then transfers that auditory information from the primary auditory cortex to this angular gyrus. Okay, and again, if it's something seen, then it comes from the occipital cortex and goes to this gyrus, to the angular gyrus. Okay, from there, the information gets transferred to Wernicke's area. Again, now, when I say transfer, relate that back to what we just learned about. Transferring means a neuron is stimulated, and that axon now there's an axon that stretches toward the Wernicke's area. So that's the axon that's been stimulated now. Okay, so that's a transfer of information. And so in Wernicke's area, those neurons now, those cell bodies, that's where the choice and the sequence of words to be spoken are formulated. Okay, so this is where you're thinking about, what do I want to say? Okay, then that information gets transferred to Broca's area. Okay, so it's like a language command that is getting transmitted to Broca's area. And so that translates this message into a programmed sound pattern. So it's determining what specific neurons need to fire, what motor neurons need to fire in order to formulate that speech pattern, to put those words together. Okay, then that gets sent toward the primary motor cortex to activate those appropriate neurons that activate the facial muscles, the tongue, the lips, right? So that desired word will be spoken about, so that you will actually make that sound, right? So Wernicke is more about comprehending what you saw or heard, and even we did put in there touch, and that's for people who might use Braille, right? So Braille can also be translated into spoken language. Okay, so again, we got to bring that in, determine what we saw and how we want to respond to it. That's Wernicke's area. Then once we determine how we're going to respond to it, we send it to Broca's area that comes up with that sequence pattern of neurons that need to fire in order to make the words come out of our mouth. Okay, the actual stimulation of those muscles then come from the primary motor cortex, and it's Broca's area that stimulates those specific neurons. So some of this we know because of people who have brain damage, and so we know that if there is brain damage to Broca's area, then they're actually not able to formulate words, right? The patient might still be able to understand what is being said to them or what they're reading, but they can't produce the words back, right? They can't speak. Damage to Wernicke's area, you can't understand the words that they see or hear, right? So they can still speak fluently, but the words will be perfectly articulated, but they don't make sense, right? They cannot attach meaning to the words or choose an appropriate word to convey their thoughts. So sometimes we refer to that as a word salad, okay? We refer to this as aphasias. Okay, so my friend, her mother had a stroke and it damaged Wernicke's area. And so I knew my friend since second grade, so I knew her mom that long. And her mom, so instead of asking us, were we going, we were teenagers, this happened to her in the third grade, when we were in the third grade. And then when we became teenagers, we'd want to go to the mall. That's what we did back in the 80s. And so we'd tell her, hey, we're going to the mall. And so she would always say, you are going to Myers. Well, Myers was like Walmart in Michigan. So we know she didn't mean Myers, but she couldn't attach the appropriate word to it. So we learned what she was trying to convey. And then if she wanted to know if we needed money, she didn't say, do you need money? She would just say, do you have dollars? And so again, we learned what she meant. And her other parts of the brain were perfectly fine. She was a very funny woman. So once I learned her sense of humor, she was very easy to get along with. Okay. Another thing to remember that it's these areas where dyslexia are associated with this. So there might be developmental abnormalities in connection with the visual and language areas. So again, all it is it's not being able to interpret what is the words that are being seen, but it has nothing at all to do with intelligence areas. So people with dyslexia, they can go through some work to learn how to read the images that they're seeing. And actually, people with dyslexia tend to be very intelligent people. We have a lot of examples of that. Okay. So what I'm going to do is I'm going to stop the recording here because that was 45 minutes of the cortex, and then I'll start a new recording and get into the other parts of the brain. So I'll see you guys in a minute.